Okay, before we start the photonic lecture series, I would like to share with you a short video. Can you hear the Okay, thank you everyone and welcome to the photonic lecture series. Okay, so the lecture today is a lecture by our speaker from Universiti UPM, Dr. Yasmin Mustapha. So before we start, let me introduce Dr. Yasmin. Okay, firstly, thank you, Dr. Yasmin, for your willingness to share with us today. Right, so Dr. Yasmin Mustapha Kamil received her Bachelor of Science degree in Biomedical Science, UKM, in 2012, and her PhD in Biomedical Engineering from UPM, Universiti Putra, Malaysia, in 2018. And her research interests include biophotonics and optical biosensors. For her PhD research, she investigated and developed a tapered optical fiber-based dengue sensor by exploring the possibilities of enhancing sensitivity through geometrical alteration of an optical fiber and incorporating nanomaterials. During her final year of her PhD, she partnered with her co-supervisor and colleague to form a company in Malaysia, Dynamic Sendirian Berhad, a startup company under the guidance of Putra Science Park UPM. And in the same year, she won the first place in the national level and Asia Pacific level of the 3-minute thesis. Today, she is a postdoctoral researcher at UPM while managing 2 small enterprises. In light of the COVID-19 pandemic, she is now looking into using tapered optical fibers as the COVID-19, the COVID-19 virus sensor. All right. So without further ado, I would like to invite Dr. Yasmin to present her sharing on optical with the title of Principle and Applications of Tapered Optical Fiber-Based Biosensors. Okay. Pastor Yumin.
Okay, let me get this slide up first. And okay, so Assalamualaikum and a very good afternoon. First and foremost, thank you very much everyone for sparing some time to listen to this sharing session. I'm Yasmin Mustapha Kamil, and today I will be talking about the principle and applications of tapered optical fiber-based biosensors. Yeah, so I hope that it's not going to be lengthy and that it's going to be brief and enjoyable for everyone, especially now that it's 3 p.m. and the last day before the long weekend. So let's get started. All right, so what is an optical biosensor? Okay, so basically an optical biosensor with the optical word in it involves light, and it should be connected to a transducer that allows light to interact with the external surrounding, and also an analyzer that can calculate this interaction. So in the presence of your targeted molecule, in this case, a dengue virus, your analyzer will be able to quantify how much that targeted molecule is in the sample. Yeah. Now, what makes a good biosensor help? So a good biosensor not only can identify the presence of the particular molecule that you want, it should also be able to quantify and should also be sensitive. Okay? And this is usually measured based on the resolution of the sensor and also the detection limit of the sensor. In other words, the lowest concentration the sensor can detect. Okay. Also, we wouldn't want our sensor to be detecting everything in the sample, so it needs to also be selective. It can distinguish between what it wants and what it doesn't want. So in the presence of a sample that has nothing of its interest, like a donut, it should be able to say that your target is not in the sample. So again, a good biosensor needs to be sensitive, where we look at the detection limit and the resolution, and it has to be selective, which usually is assessed using a negative control. Okay. So today I will be introducing to you a unique transducer. It's a transducer that's fabricated from an optical fiber that's being pulled to gain a narrow diameter in a particular region. And we call this a tapered optical fiber. So in order to appreciate this technology, I will be giving some refreshing description on the theoretical concepts of the tapered optical fiber sensor, how we fabricate them, and how we develop them in making them into a biosensor that is selective, and how we enhance the sensitivity. And last but not least, I will discuss a bit on the advantages and disadvantages of this transducer. All right, so we're really going back to basics. We know that light is the fastest in a vacuum, right? So, but once it comes into a particular medium, say water, the speed reduces because now there are particles in the water, in the medium. And when you compare these two light speeds to, you will get this parameter known as a refractive index, which is important in this case because it governs the way how the sensor works. In other words, a refractive index shows the speed of light within a particular medium. So if the refractive index is higher, the speed of light in that particular medium is slower. So that is why air, we get 1 as a refractive index, or 1.0003, and water, it's about 1.322. So then what happens when light passes between two particular mediums, say water into air. So in Snell's law, we know that the ratio of the refractive index between the two medium is somewhat the same to the angle of refraction to the angle of incidence. Okay, and at one point, at a particular angle of incidence, the refracted angle will become 90 degrees, and at this point, we call the incident angle as the critical angle. This is important because in order to contain light within a particular medium, we need reflection to occur. And reflection can only occur when the incident of light is at the angle of the incident of light is larger than the critical angle, which can be achieved by manipulating the incident angle or the refractive index of both mediums, which is what happens in optical fibers. So an optical fiber has two major layers: the core, where the fundamental modes or where most of the light is concentrated, okay, and the cladding. Okay, so how you get light propagating from one distance to another is by manipulating the refractive index of these two layers. So in an optical fiber, say in a single mode optical fiber in general, you should have the refractive index of the core higher than the refractive index of the cladding, so that light will be reflected. Will be reflected within the core with minimal loss. Okay? So then what happens in a tapered optical fiber, where you hold the fiber to get the narrow region in the middle? Well, first, you'll get three different regions: the down taper, the taper waist, and the up taper. And then here are some important parameters in a tapered optical fiber, which is the taper angle and also the core diameter. So when light approaches the down taper region, due to the reduction of the core diameter and also the angle, taper angle, we get light modes within the core we get excitation of the light modes. Okay? And due to this, some will escape from the core and cladding boundary into the external medium, especially when it starts to get very, very thin. Okay, so when it starts to get very thin, say, we usually taper down our taper fibers to 10 microns as a whole. So for example, from 1 to 5 microns to 10 microns. The layers are now too thin that the whole core and cladding becomes the core and the external surrounding becomes the cladding. And when this happens, you have some light at the boundary of the tapered fiber and also the external medium. And this is what we call evanescent waves. Okay. In simulation, okay, you can see that as the core reduces, the intensity of light that escapes from the core becomes bigger. So, which is prominent here. So, yeah, that's what happens within the core of the tapered optical fiber. And as the smaller the diameter goes, the larger the evanescent field becomes, or the steeper the taper angle is, the larger the evanescent field becomes. And when you reach to the upper taper, the light that's propagating on the outside of the fiber will couple back into the fundamental mode. Okay, and this is the resulting transmission spectrum that you will get. So with a normal optical fiber that has not been tapered, when you show broadband light source within it, you will get a broad spectrum. However, with a tapered optical fiber, you will get a fringing pattern. And this is because of the coupling between modes. And this is in a single mode fiber. So in a multi-mode, it will show a completely different pattern. But so this shows the coupling of modes. That becomes the fringing pattern. Okay. So why is this important? Well, we want some light in a sensor. We want some light to escape from the fibers so that it can interact with whatever that is surrounding it, and then couple back. And then the changes will be quantifiable based on the optical output that you get in the end, which I will explain more later. So let's go to the fabrication of the tapered optical fiber. So as I mentioned, it's a heat and cool technique. And the heat source can be through a flame. People usually use hydrogen flame. And there's a technique called flame brushing to get a homogeneous length. Some will use an electrical heater and a laser heated tube as well, like a CO2 laser. At our lab, we have a glass processing machine known as the Vitrin machine that allows us to get tapered optical fibers based on the parameters that we want. Okay? And the heating source is a graphite filament that is being heated up at the furnace, so that when the rig starts to pull the tapered fiber, you will get a narrow region where it's being heated up. So here's a video of how it works. Yeah. So once the heating thing happens, the rig will pull the fiber, and then you'll get a narrow region. Okay. So now we have fabricated our tapered optical fibers. How do we make it into a biological sensor? Okay. First, we need to identify the biological targets that we want. So usually people would want to detect viruses, the DNA, RNA of particular virus or a bacteria, metabolites like or antibodies, enzymes, and bacteria. Okay. But then now, if it's just being thrown like that, the optical fiber won't be able to recognize or distinguish what it actually wants. So then selectivity won't be there. So how do we get the selectivity for the tapered optical fiber? By using biorecognition molecules. In other words, biorecognition molecules are molecules that recognize the target specifically. For example, we can use enzymes that are specifically, has a specific affinity to a particular particle that we want, or an antibody to an antigen. So in the dengue sensor that I was working on, the biorecognition molecule that we used were antibodies to detect the E protein of the dengue sensor. So how do we get these proteins on the fiber? First, there are some surface functionalization steps that we have to do. So first is the hydroxylation of fiber. Here we use sodium hydroxide, and this is to get hydroxyl groups on the surface of the fiber. To facilitate the next step, which is the selenization. And this is key because we need silane agents to get organic molecules onto an inorganic substrate like the glass, like the tapered optical fiber. And here we use a chemical called uptest. And once we've selenized the tapered optical fiber, we move on to the activation, where we introduce it to glutaraldehyde, and this is to enhance the protein support interaction. And to avoid steric effects and conformational changes. If we do not have glutaraldehyde, the distance between the antibody and the surface is too close. And because of that, we would get some conformational changes and the interaction won't be optimum. So that's why we needed glutaraldehyde. And after that is our biorecognition molecule, which in this case is the antibody to the dengue virus. So let's say if you're interested in developing a biosensor, this is where you put your biorecognition molecule, which can be an enzyme or whichever that has high affinity to your targeted molecule. Okay. So these were taken by my colleague, And he was working on a dengue sensor as well, but using multi-mode tapered fiber and looking at the absorbance. So so here are pictures of functionalized tapered optical fiber. And as you can see, the small circles here are actually the dengue virus themselves. So in his project, he detected the virus itself, unlike mine, was just one protein of the virus. So yeah, nice pictures, right? So then once we have functionalized the optical fiber, how do we detect our targeted molecule? Well, we can look at a number of optical parameters. In my work, I look at the wavelength shift, okay? And in like my colleague's work, he was looking at absorbance. We can also look at fluorescence as well, but all of these things depend on the mechanism of the sensor that you're building. I'll give you some examples. Okay, for example, if we're looking at wavelength shift, this was the experimental setup that I used. So it's pretty simple. We need a broadband light source, a C-band light source, and then it goes through a sample holder where the tapered fiber is situated in. That is where I will introduce the solutions and the sample. And then it's connected to an optical spectrum analyzer. All right. So remember in the beginning, I mentioned that because of the two modes colliding, coupling, we will get a fringing pattern. Well, when the refractive index of the external surrounding increases, this spectrum will shift based on these two equations. Okay? So it will shift to the right, to the higher wavelength. Okay? So as the concentration increases, the refractive index also increases. So from there, we can quantify and we can relate the wavelength shift to the concentration of the sample. Okay? So at our lab, as what I was working on for the dengue E proteins, we managed to get a sensitivity of 5.02 nanometer per nanomolar with a detection limit of 1 picomolar. I have another colleague who worked on leptospira DNA. So she detected, her target molecule was DNA, and her biorecognition molecule was a DNA probe, and her sensitivity was 1.29 nanometer per nanomolar with the detection limit of 1 femtomolar. So that's pretty low detection limit. In other words, good. And a plus for tapered optic fibers that it's very strain sensitive. Okay, so when we look at the absorbance instead of the wavelength shift, this will usually be the setup that people would use. So there will be a light source. Usually it's a tungsten halogen light source. And then it will go to a tapered optical fiber. Considering we're looking at absorbance, most researchers would use multi-mode fiber. Okay? And then it will be read using a spectrometer. So in this study that I would love to share with you guys, they have actually customized their own sample holder as a microfluidic chip. Okay? And they employ the optical fiber, the tapered optical fiber within the microchannel. Okay. So what was he trying to do? He was trying to detect bovine serum albumin. It's a common protein, BSA. And how does he do that? Well, there's this famous assay known as the Bradford assay, where it's a protein assay to calculate the protein concentrations. So basically it uses a chromogen. So the Bradford assay is usually brown in color, brown to red, but with the presence of protein, it will turn into blue due to some electron transfers. Okay, so that was the mechanism that he used, and the concept is that with higher concentrations of BSA, he is expecting to see higher absorbance at 595 nanometer to about 650. So that's what he got here. As the concentration increases, he will get larger absorbance. So yeah, so this is how you look at absorbance instead of wavelength shift. It all depends on your mechanism. And the detection limit for his work is 10 femtogram per mil. That's also quite low. Another way to assess is using fluorescence. To me, this is pretty interesting. So the concept here uses fluorescent resonance energy transfer, or also known as FRET. Basically, what it does is you have a fluorescence donor that fluoresces, okay, and then you have a fluorescent quencher that somehow absorbs this fluorescence, okay. So they have those two elements here. What these people did was, first they have the partially reduced graphene oxide as the quencher, and they deposit that on the tapered optical fiber. And then they coated it with the donor, which is rhodamine 6G. Okay? So when that happens, you will get a fluorescent signal that is low, because rhodamine here, despite it fluorescing a particular wavelength, we have the PRGO quenching it, right? Okay. So then what happens is, when the targeted analyte is being introduced, there will be a competition of binding that occurs, which will somehow kick out the rhodamine from the PRGO, and it binds onto the PRGO instead. So then when rhodamine is free, the fluorescence will increase. Okay, I hope you get that. So what happens here is they were trying to detect 3 different targeted molecules: cadmium ion, dopamine, and single-strand DNA. And they made their sensor selective based on certain pre-treatment processes. Okay? So for the first type, it was direct without any pre-treatment, and that was to detect cadmium. For the second type, it was pre-immersed with nitrate. And for the third type, it was first pre-functionalized with Na+. Sorry, this shouldn't be capital, but yeah, Na+ ions. Okay. So if you look at the results, each type is selective to the targeted molecule that it was aiming for, thanks to the pre-treatments that they did. Okay. So, for example, type 1 was to detect cadmium. You can see here the fluorescence is higher when introduced with cadmium compared to the rest. And it goes the same for dopamine and also SSDA with respect to the type of the fiber. So that's how you can assess your biology sensor based on its fluorescence. Okay, so now that we have some idea on how to assess our sensor, we go with how to enhance the performance of our sensor. Okay, so in my work, what we did was we incorporated nanomaterials. Among what was tested was graphene oxide. And the reason why was because graphene oxide is a single layer hexagonal lattice, and it has a high surface to volume ratio, and it is decorated with many hydroxyl groups. Okay? And this will promote more binding sites for immobilization to occur. And this is good because if we have more of the biorecognition molecules on the fiber, then we have more chances of getting the targeted molecule. Okay. So it has been used in past studies, for example, for an Alzheimer's mRNA markers with a detection limit of 1 picomolar. It was also used in ethanol sensing that gave 2 times the better of a response. and also for volatile organic compounds, which also showed better sensitivity compared to reduced graphene oxide that has lesser hydroxyl groups on the lattice. So when we used it in our tapered optical fiber, how we functionalized it was from the very beginning. So we deposited the GO first, and then we start the surface functionalization steps on the GO. Of course, when we coat with nanomaterials, thickness plays a huge role. So we tried different thickness by manipulating the immersion time. And out of the thickness that we tried, the best was with immersion time of 30 minutes. That gave 12.77 sensitivity. Sorry, 12.77 nanometer per nanomolar, which is.
I'm not sure.
Yeah.
Any question from somebody raised her hand?
Oh yeah, sorry.
Sorry to disturb you, Min.
It's okay.
I think just now your voice there is some disturbance. I'm not sure because of your mic. Cuba Min mic tu guna external mic
Tak. Laptop.
Kenapa? Basically, you can just now, Sarah, do you have any question? But maybe kita tekan kot. Sorry to disturb you.
It's okay.
Maybe Min kena duduk dekat sini ke? Sometimes
tak dengar.
your voice is not that clear.
Sorry. Okay, better tak?
Better. Sorry to disturb you. You can continue.
Okay, so as I mentioned just now, the thickness of the nanomaterial is important, and we tried a few thicknesses, and we managed to get the best at 30 minutes of immersion time, which yielded a sensitivity of 12.77 nanometer per nanomolar, which is more than double the sensitivity without graphene oxide. So that's good. So that's how we enhance the sensor with graphene oxide. We also tried another nanomaterial known as palmum dendrimer. Okay, reason being is because one, it is branch-like and it's stable. Branch-like. Why do we like branch-like? Well, now that if you look at the molecular structure here, there are so many amino groups, right? To enhance the binding of the biorecognition molecules. So that's what we want. And in previous studies, we have seen that palmum has been used for pH sensing in cells. We have also seen it in SPR immunosensor for TNT, and also protein loading on U-shaped fiber that gave a 2.2-fold of sensing performance. The same we tried with different thicknesses as well. And however, because of the difference in molecular structure, the way we deposited it was not from the very beginning, because now it's an organic material, right? So it needs to be selenized first. I mean, the tapered optical fiber needs to go through selenization first, and then the activation, and after that we use palmum. And on top of that, the biorecognition molecules. And the best performance yielded at immersion time of 40 minutes that gave 19.53 nanometer per nanomolar sensitivity, which is 3 folds, close to 4 folds of the sensitivity that was attained with the normal configuration. Reason being is one, the structure of it being branch-like, and two, because the functional groups on it is amino groups, it somehow improves the orientation of the antibodies, and it exposes the active sites of the antibodies outwards, sort of, so that it's easier for the targeted molecule to react with it. So yeah. And when we compare, so I've also tried combining both graphene oxide and palmum. since both enhance the performance, why don't we just put both, right? But Palmum, the sensitivity attained with Palmum is better still. I would say it's because of thickness as well. We did not fully optimize the thickness when we combine it. We just used the best thickness for both individually and then combined. So if anyone's interested in looking into this, optimizing the thickness is the way to go. Yeah, you might get better sensitivity. Okay, and so we also look at the dissociation constant. Apologies, this shouldn't be m-1, it should just be m. Now, dissociation constant shows how easily the targeted molecule and the sensor dissociates. Yeah, so so it's a dissociation rate. Yeah, so we wouldn't want it to dissociate easily, right? Because or else that means the affinity of the sensor is not high towards the targeted molecule. So that's why we look into the dissociation constant as well. All right, so now that we have already looked at the enhancement of the sensor, let's discuss a bit on the advantages and disadvantages of tapered optical fiber, and in a way to conclude this sharing session. So after years of working with tapered optical fibers, I have to acknowledge that the advantages would be one, it's a seamless system. In other words, it's an all fiber system, so it's seamless. And it can promote array sensing as well. In other words, say if we have one tapered fiber and then we taper different regions, right? And let's say each region is sensitive to a different targeted molecule, so we can get like an array of sensing in one system. And also it's small in size. It is modifiable for various sensing mechanisms. It is highly sensitive, and because of its minute in size, it only requires little volume of sample per sapling. And in the healthcare field, that is important, because we wouldn't want to need a lot of samples to go through one test, right? Now, the disadvantage of an optical fiber is that one, it is too fragile. So you require gentle hands to handle them. I remember being very disappointed in the very first 3 months or 4 months of my studies in the beginning because it keeps on breaking. So and also another disadvantage is that it can be too sensitive to just anything within their surrounding, despite it being selective. So, for example, you know how we fix it onto the sample holder. If there is a slight tinge of change in pressure, let's say the volume of your sample is not the same, your fiber is hanging at the top of the sample, you have surface tension there, how you pull or fix the fiber onto the sample holder, there's some changes in pressure there as well. You will get differences in your results, because it also contributes to so it also affects the optical properties in the end. So so that particularly wraps up my sharing session. To recap, I have the aim today is basically to introduce to you guys a tapered optical fiber sensor and the basic principles of it, how we develop it into a biosensor, how we can assess it in different ways. It all depends on your creativity and how we enhance the sensing performance. So I hope that this short sharing session will be able to give you guys some ideas if some ideas on how to improve, how to develop sensors, with using a tapered optical fiber. So and with that, I end my sharing session. Here are the pictures of the members at our lab. As you can see, all smiles here because it's a happy place. But yeah, if you guys are interested in collaborating with our lab, send me an email. We don't only do sensors. We do lasers as well. And yeah, that's about it. Thank you very much.
Thank you, Dr. Yasmin. There are a few questions. One question from Dr. Nadia. Dr. Nadia asking, what does array sensing mean in this case? Actually, yes, Dr. Yasmin also can see the question in the chat box.
Okay, so thanks, Dr. Nadia, for your question. Array sensing means you can sense a number of targeted molecules at once within the same system. So as how I've explained just now, if we have different tapered regions within one long length of optical fiber, and it's targeting different molecules. So you will be able to detect different targeted molecules within one system. So that's what an array sensing means. And how this being too sensitive become a limitation? Let's say, despite the sensor being selective, as I mentioned just now, despite it only targeting our, dengue virus, right? It's still sensitive to other parameters as well. For example, like temperature, temperature and tension. So, and it's too sensitive that sometimes it becomes noisy. And that's a limitation because then it's difficult for you to distinguish if that shift comes from your targeted molecule or is it coming from surrounding changes? Changes within the surrounding. Yeah. So that answers, I hope that answers your question, Dr. Nadia. Should I just continue answering questions?
Yeah, you can continue. The next question is from Nishat.
Okay, so Nishat, hello. I just would like to know the approximate diameter of the waist in your taper fiber. Okay, so in the very beginning of how we started, we did an optimization of the tapered optical fiber parameters. This includes the up taper length, the waist length, and also the down taper length. In my work, we use a core diameter of 12 micrometers. Yeah. So from 125 micrometers, downsize it to 12 micrometers. Why? Somehow when we tested different parameters with different concentrations of NaCl, sodium chloride, the parameter that I'm using somehow yielded the best sensing performance and also the most feasible one. Because if we go too low, let's say a waist of 3 microns, it will be too fragile to functionalize anything and to handle. So that has to take into consideration as well. Unless if you have other alternatives on how to handle it, that would be great. However, at that time we do not have any alternatives to handle a very, very small diameter. So yeah. Next question is from Dr. Akmal Yahya. How do you measure the thickness of the nanomaterial coated? Seems the thickness is super thin, less than 20 nanometer thickness. Okay, so we use AFM. So yeah, we measure it using AFM. I hope that answers your question. Okay, next question is from Sa'ad. Considering the fragility and sensitivity to slight changes, what can you say about packaging challenges for practical application? Question. Let's not go to packaging. Let's talk about lifetime, right? There are challenges before packaging itself. When we look at lifetime, imagine how do we even assess lifetime and ensure that nothing gets interferes with it within the lifetime. So what can you say about packaging challenges? I can say that it will be very challenging. That's what I would say. However, if we can figure out a way to somehow control the condition in terms of how we fix the fiber, in terms of temperature, and bear in mind that we functionalize proteins onto the tapered fiber. The temperature, it becomes temperature sensitive, right? So all of this has to be taken into consideration. Yeah. Okay. So next is from Nora's Riena. Hi, Dr. Yasmin. You mentioned that the thickness of the tapered fiber is important. How about the length of the taper waist? Okay. So when we were looking at different taper parameters, when everything else is being constant, and when we manipulate the taper waist, which in the end will be the interaction length, the longer the waist, the more fringes you will get, and the smaller FSR you will get. Is it important? Yes, it's important, but maybe not to a degree of how the thickness of the core is. However, it is important because it does affect the spectrum as well. So that needs to be considered too. Okay, next is Dr. Nazira Mohamed Razali. Does the transition part of the tapered fiber affects the sensor performance? Yes, because the transition part is what determines your taper angle. Okay? So in the beginning, I did mention that if the diameter of the taper fiber is smaller, the smaller it gets, the more evanescent wave evanescent waves you will get. The reason being is because you will get a steeper angle, right? With a smaller diameter and a bigger diameter. So it's all about manipulating the taper angle, where the transition part plays a role as well. So the transition is important. However, in my work, we did not look at the transition length as much. We maintain it as constant while we were playing with the other parameters. So ultimately, whatever that affects the taper angle will cause a change in the sensing performance. Okay, so then Norazrina asks again, may I know the repeatability of your sensor? Okay, the sensor that we developed is a one-off sensor. Okay, so meaning it can only be used once. But however, we did support the repeatability by making different sets of the sensor and by comparing the results. So in terms of percentage, it will be close to 80%, I would say, in terms of repeatability. Yeah. And I guess that's any more questions? Okay, so Nora is asking, may I know the best method to deposit your tapered fiber with nanomaterial? It depends on your nanomaterial, and for graphene oxide, I wouldn't say it's the best method because I did not explore other deposition methods. But what we did was a drop cast. We used the drop cast method. So we just basically immerse the tapered fiber within a pool of the graphene oxide. So yeah, same goes with palm. We just immerse the tapered fiber in the palm solution. So I could not really give you the best method because we have not experimented other methods. So, but that was what we did. Okay. Any more questions?
Maybe that's it. Yeah, any more questions before we end our session? If no more questions, so I would like to thank Dr. Yasmin for sharing on the tapered optical fiber sensor today. Thank you very much. And before we end our session, I would like to get a photo, to take a photo, group photo. Min, can you stop sharing your slide?
Oh, yes.
Yeah. And if possible, if everybody can turn on their camera, we can have the photo. If you want to put a background, if you can also set some background if you want. Yeah. Okay, before that, let me change the layout. Okay. Hold on. Okay, everyone. I will count until 10. Please smile. Okay, 1, 2, 3. Okay, one more. 1, 2, 3. Okay, thank you. Thank you everyone for joining us today. Okay, and also don't forget to fill up the attendance through the link that I share in the chat box. See you again in the next Photonic lecture series. And if you want to know more regarding IEEE Photonic Society, the activities that we have, just email us. Okay, and you can contact us. You can contact and ask me or any IEEE Photonic society committee. Right. See you again in the next Photonic lecture series. Bye bye. Thank you, everyone.
Thank you.
Thank you.
Thank you, Dr. Yasmin. Be safe. Take care.